Hi, my name is Dr. Scott Coffin. I am a research scientist with the California Office of Environmental Health Hazard Assessment, and I'll be talking to you today about California's adaptive risk management of microplastics in aquatic ecosystems and drinking water. In 2018, the California Water Boards received a mandate from the legislature to address microplastics in drinking water, including defining microplastics and adopting a standard method for monitoring, a plan for 4 years of testing and analysis, consider adopting a health-based guidance level, as well as accredit qualified laboratories for testing. The same year, California Senate Bill 1263 was passed, which requires the Ocean Protection Council and the California State Water Boards to initiate a statewide microplastics strategy by 2022. Included in the strategy is the plan to develop a risk assessment framework, standardized analytical methods for monitoring, establishing baseline occurrence data of microplastics in aquatic ecosystems, and figuring out how those particles are getting into our ecosystems, then recommending source reduction strategies. A key component of both of these bills is developing a standardized analytical method for monitoring and for sampling of microplastics, as well as accrediting qualified laboratories. When these bills were passed, there were no standardized analytical methods for microplastics. So we developed our own and validated their performance with the help of 26 volunteer laboratories from 7 countries, as well as 2 methods using FTIR or Raman spectroscopy. We developed this method not only for drinking water, but also much more difficult matrices, including ocean water, fish tissue, and sediment. We evaluated the method performance as well as the strengths and weaknesses of these different methods in terms of their cost, as well as how long they take to run. And we found that it's necessary to use a spectroscopic technique to be certain that the particles you're finding are indeed microplastics. With just guessing at the particle under an optical microscope gets you about a 44% accuracy rate. Infrared and Raman spectroscopy are not cheap instruments, though, costing between $550 and $337,000, depending on the type of instrument. The consumables for these instruments are also non-negligible, and what's really important to understand is that these methods can take a very long time, especially if you do not use automated techniques. An average of 10 hours per sample for infrared and 15 hours per sample for Raman. Laboratories who wish to become accredited for one of our 2 standardized analytical methods can do so through the Environmental Laboratory Accreditation Program, or ELAP, which is part of the State Water Resources Control Board. Note that laboratories need not be based in California. They can be based in any part of the world and be from any sector. A key component of what we found in our study is that we need not use one method for all types of analyses, as Raman and infrared and the other available methods in the literature are complementary in the types of particles that they can reliably detect in terms of size, the color, as well as the polymer type, and different methods may be used in different circumstances. An example of how this might be done in practice is through our proposed tiered monitoring framework, where one could use a fast and inexpensive tool such as Nile red fluorescence to identify likely plastic particles in certain samples. And then above a certain threshold, they would then go and use a more targeted method such as pyrolysis GCMS to get the mass of the plastic particles, or using our FTIR and Raman. This framework is very similar to another tiered monitoring framework that has recently been developed for per and polyfluoroalkylated substances, or PFAS. Next, I'll be speaking about what we did to evaluate the health effects of microplastics, both for humans through drinking water and for aquatic ecosystems. To evaluate the health impacts of microplastics, we convened an expert workshop and screened the literature, asking these key questions. Which dose metrics predict toxicity? Are there certain particle characteristics associated with harm? What are the adverse effects and can we predict them? And do we have enough information to come up with a threshold or a threshold framework for regulatory guidance? Our expert work group identified 3 main ways that microplastics can be hazardous to both humans and aquatic organisms, including the chemicals that are bound to these plastics, biological means, as well as particle-based means of toxicity. There are over 10,000 known chemical additives used in plastics, of which 2,400 are considered to be hazardous. Unfortunately, 53% of those 2,400 are currently unregulated in one or more parts of the world, and we lack scientific references for approximately 11% of those toxic substances. In addition to chemical additives, microplastics can also act as a vector for pathogens as well as an enrichment hub for antibiotic resistant bacteria. We've found that the co-exposure of microplastics with certain viruses can cause excess mortality, more than just the virus or the microplastic particle alone to select organisms. While we are indeed concerned about the chemical and pathogen-based hazards of microplastics, the most reliable evidence that our expert group found was for the particles being toxic themselves. For the evaluation of human health impacts via drinking water, we found 29 ingestion-based in vivo mammalian microplastic toxicity studies, of which 12 were deemed fit for purpose for expert review. The most reliable evidence that we found from these 12 studies dealt with male reproductive impacts, all in rodents. We found consistent impacts across multiple studies, with 4 different studies showing negative impacts on sperm deformity, 2 on sperm count, and 2 on lower testosterone, with some additional studies pointing to male reproductive impacts occurring from these particles. We also found 2 reliable studies indicating potential harm to the female reproductive system, two that affected body weight, and 1 study showing impacts to the liver. Despite this evidence, our expert work group was not able to derive levels for regulatory use. This is due to the lack of a adequate effects database. In particular, most of these studies were not doing a very good job at characterizing the particles they exposed the organisms to. They were also using a very limited subset of polymers, shapes, and sizes, mostly polystyrene spheres around 5 micrometers in diameter. We also are uncertain regarding the effect mechanisms of these particles, which is necessary for the extrapolation to the diverse particle types that we are all exposed to. And finally, we have an incomplete exposure data set with limited information on what's in our food and no harmonized drinking water data that we can rely upon. In terms of assessing risks to aquatic organisms, here we were dealing with far greater number of studies, 167 in total, with documented effects on 117 species. Unfortunately, the vast majority of these studies were not deemed to be reliable according to our quality standards. Of these, only 22 studies and 16 species were considered to be useful, which then underwent additional expert review. We then did a little bit of data alignment to make sure that we could compare apples to apples, so to speak, and then performed what's called species sensitivity distribution modeling to estimate harm to aquatic ecosystems. A species sensitivity distribution looks like this, where you line up your species based on the concentration that affects them, with the most sensitive species on the lower end of the curve and the least sensitive species on the high end of the curve. With modeling, you can then predict the concentration that would cause an impact to 5% of species, for instance, or a different value that one could select. Using this approach, our expert work group derived toxicity thresholds corresponding to 4 different management levels of concern and action. At the lowest level here, we would recommend investigative monitoring to further investigate the issue, with the highest threshold considered to be more certain in terms of the severity and the likelihood of impacts occurring to the ecosystem, in which case we would recommend that source control measures be taken. We derived a set of thresholds for a common mechanism for which we have reliable data, which is food dilution, in which the particles are taking up space in the organism's gut so they can't get food from other sources, as well as tissue translocation mediated impacts, whereby the particles are accumulating in the tissue of these organisms and causing cytotoxicity and inflammation. It's important to note that the thresholds that I just showed you are applicable to the types of microplastics that we see globally in aquatic ecosystems. And the particles that may occur in your specific water body of interest might look quite different. in which case the thresholds are going to appear quite different. You may also be interested in different types of organisms for your specific ecosystem. So we made a tool publicly available that I'm showing here. This is the Toxicity of Microplastics Explore application, which allows one to derive custom thresholds for an aquatic ecosystem of interest. Showing here just how quickly one can filter and select the values, as well as align the particle-based toxicity thresholds to your specific ecosystem. And boom, you have your own species sensitivity distribution that you can then use to assess risk. In addition to being a data visualization and threshold development tool, ToMEx is also a living database of toxicity information for microplastics for which anyone can upload published data to that will then be reviewed by external reviewers. Currently, we are working on updating this with a large group of collaborators, and for ToMEx 2.0, we've added 22 additional studies for human health, as well as 125 new studies for aquatic organisms. This brings the total data set up to 12,500 data points for in vivo toxicity studies for aquatic organisms, making it by far the largest repository of information for microplastics on toxicity. Now I'll talk briefly about what the State Water Board is doing for its plan for 4 years of testing and analysis of microplastics in drinking water. Microplastics can make their way into drinking water sources through many routes, including terrestrial emissions, atmospheric deposition, and emissions from municipal and industrial wastewater effluent. Once microplastics make their way into a drinking water treatment plant, they may be removed by the treatment processes. For instance, flocculation and sedimentation are going to remove microplastics down to about 5 microns or so, and our analytical methods are only reliable down to about 20 to 50 microns, depending on the technique. This means that if we were to look for microplastics in water that's been treated using one of these techniques, it's unlikely that we'll be able to actually detect those particles. However, there still may be particles that are getting to the consumer, and especially the particles that are smaller than 10 micrometers. These are the ones that we consider to be toxic to human health because they can translocate tissue and accumulate in our organs. Understanding this limitation of our standardized methods led us to the development of an iterative monitoring approach for testing microplastics in California's drinking water supplies, in which we are going to mostly be focusing on the source waters for the first 2 years, it's shown in phase 1, and looking for microplastics in treated drinking water starting in about 2027 or so, once we have additional, more sophisticated methods that might be able to get those particles that are breaking through the treatment processes. A key aspect of this monitoring campaign is ensuring the data we receive is reliable and interoperable, which is why we developed an open source harmonization portal and database called One for All, which ensures that laboratories only submit data that meets California's standards. I'd like to talk briefly about how we applied the aquatic ecosystem thresholds to monitoring data received in San Francisco Bay, California. We compared surface water monitoring results of microplastics in San Francisco Bay to our aquatic ecosystem thresholds and found that the majority of samples within San Francisco Bay exceeded the investigative monitoring threshold, suggesting that there may be potential risk occurring in this ecosystem. The State Water Board is currently evaluating this risk assessment and determining whether or not we should place any of these water bodies on the Clean Water Act's 305B list, meaning that there's not enough information to determine beneficial use support, but there is information that indicates beneficial uses may be threatened. To meet the requirement of Senate Bill 1263 to develop a statewide microplastic strategy, California's Ocean Protection Council adopted a two-track approach in 2022 involving solutions that no-regret solutions that can be taken today, such as pollution prevention, pathway intervention, and outreach and education, as well as science to inform future action, including monitoring, developing risk assessment thresholds, evaluating sources and pathways, and evaluating new solutions. None of this work could have been accomplished without the microplastic community's commitment to free and open sharing of data, tools, and resources. If you'd like to learn more about these open resources, you can find them on a website I created called plastiverse.org. Thank you all for your attention. I look forward to connecting with you further, and happy Earth Day.